Good morning. This morning we're going to have, again, two presentations. The start will be by Meredith Fitzgerald, who's the director of the Clinical Research Support Program. So the CCTS actually, Meredith will give you some more detailed information about this, but offers services that really are designed to allow people who don't have a large clinical trials kind of operation or a clinical research operation to have access to all the tools they need to do their research. So you can rent space, you can pay for part of the time of a study coordinator to help you with things. There's all kinds of things that CRISP offers as support for people who want to do human studies-based research. And so she'll be talking about that, but the primary focus of what she's going to talk about is the concept of good clinical practice, which is a systematic way to make sure that people are prepared to do research with human subjects in a ethically correct and strong way. The second part of the session will be by Dr. Jeff Edberg, who runs a clinical sample processing core as well as a biorepository for samples, and we'll describe some of the key issues around the function of those units and how to access those as well. Next week, we'll have the final part of our bioethics component of this course. Dr. Tom Huddle will be talking about some of the more subtle aspects of human studies research ethics, and it's a pretty interactive discussion, so get woken up before you come and prepare to speak. So thanks very much, Meredith. Glad to have you.
So thank you for having me. What we do in the Clinical Research Support Program is, like Dr. Chaplin said, we have staff that are well-trained and credentialed across campus. So they can go to Children's Hospital, they can go to all over UAB, on campus, and in the medical facilities. And so if you're an investigator writing the protocol, we can give you some feedback on how to implement. We're more of an implementation team. We have nurses and non-nurses. We have both that are skilled in phlebotomy. Our nurses are skilled in basic nursing skills. A lot of them have extensive clinical or clinical backgrounds in different areas, and we try to match the areas to whatever you're doing. We also have regulatory people, and they will do anything from your IRB application to OSP. And then we have budget building capabilities. So we'll go in, sit down with your staff, assess, do a feasibility assessment, and we will help you develop your budgets, whether you're doing a grant or whether you're doing a pharmaceutical study or behavioral study. It really doesn't matter. We try to help you with that. And then we have data entry and specimen. So basically, anybody can come to us with anything that they need, and we try to fulfill those needs. So we see everything across campus. And so when I do this presentation, I try to draw in real-life experiences to try and make it a little more applicable and more interesting. GCP is probably the most, or good clinical practices, is the most important part of research. It's in everything. So basically it's the building blocks, not the most fun, exciting topic, but really essentially important to everything that we do. So this is, our objectives are to explain the why, the what, the when, and the who. So what is the meaning and the intent of GCPs? So if you take anything out of this presentation, this is the most important slide. It's to provide guidelines to assure the rights of study subjects are protected in research. That's the most important thing. Your subjects' safety is the most important thing. And to provide guidelines to assure that the accurate and reliable collection of data in research. So what is research? It's a systematic investigation, including research development, testing, evaluation, designed to develop or contribute to generalizable knowledge. So basically, it's systematic. Your protocol defines exactly what you're supposed to do. You follow it, and you do the same thing, so it's generalizable, and you can reproduce it. So how do we protect human subjects? Probably one of the most important things, and a lot of times we overlook when we're writing protocols, is the design of the study. A lot of times we're so focused on the scientific part of it, we forget about how are we going to implement it, how are we going to do it. So it's really important that we think through the whole process. And we design the study with safety and how to collect the data in mind. And what data you're collecting. I sit down with a lot of investigators and they haven't even thought through what they're going to collect or how they're going to collect it or what they're going to put it in, what kind of database and stuff like that. The review of the local IRB, the Institutional Review Board, that they're here to help make sure that it's safe. Whether you're doing behavioral research or you're doing a clinical trial or a device trial, anything. And the protocol defines safety measures. When I read a protocol, I really go in and look at what my outcomes are, what my AEs are, adverse events, severe adverse events could be. And then I really pay attention to how that can affect my subjects. And I think what I want my mom and dad to do this. If it's a behavioral study, we take for granted and we'll say, oh, it's just a behavioral study. We're just going to ask a couple of questionnaires on how they behave or what they do in their daily lives. But actually, questionnaires can cause a lot of stress. The IRB likes to give an example of going to the clinic and saying, okay, I need everybody that has hepatitis. Well, of course, you know, that A, singles out a population, and B, all they're going to do is administer a questionnaire. So that can be quite stressful if they're answering personal questions about their life. Another example is behavioral studies. Somebody from education going into a class and they want to study what obese children eat on a daily basis. So all they want to do is question the obese children. Well, that's not right either, because then you're singling out a population and the other students are noticing that you're just looking for this population. So a lot of times we don't think about this, but that's what the IRB is there for, is to make sure that we're not jeopardizing the safety of the subjects or stressing them out. And we need to make sure our research staff is trained. Regular communication, I can't stress this enough. In directing Chris, we see a lot of different research. We see a lot of different investigators. And basically we see the good and we see the bad. And one of the things that I see with some of the areas that aren't doing it up to par is that there's not communication between the investigator and the research team, and they're not trained. We had an investigator from the FDA come and talk, and one of the things that she looks for is that you're well trained. So it's real important that you have training logs and things, and that you're not a dermatologist doing CV surgery or something like that. So what are the International Conference on Harmonization, or ICH, is what I like to refer to GCPs. So internationally agreed upon quality standards that can be used by governments in the creation of regulations for clinical trials involving human subjects. So basically, they outline the responsibilities and expectations of everybody conducting the research. Whether you're an investigator, you're a monitor, you're a sponsor, IRB, they give you good outlines. GCPs cover all aspects of research, from monitoring to reporting to archiving, data collection, all of those kind of things. So whom do the GCPs apply? Everybody. From the investigator to the secretary calling to schedule an appointment for a patient, for somebody that's administering a questionnaire. Any person that is in contact in that research team or with that patient is part of the team, and they apply. The goals, again, can you see the message that we're stressing? To protect human subjects and make sure that the data is reliable and accurate. And the impact of it is, if you do this from day one, it strengthens the ethical and scientific standards that, when adhered to, enhance the quality of research. Because why are we doing it? They were developed with multiple countries, in accordance with multiple countries and the World Health Organization. So where do they come from? Unfortunately, we had some investigators some public tragedies that involve serious fraud and abuse of human rights. I mean, you may be familiar with the syphilis trial, Tuskegee, Jesse Gessinger. There's numerous ones. And just recently in, I want to say, north central Alabama, there was an investigator that wrote and filled out every one of his consents and drew blood from his office staff. He's now in jail, but it still happens today. And I mean, unfortunately, so it does, and we have to be aware of it, and we have to be diligent in how we do conduct our research. So from these tragedies, they develop these documents and regulations to make sure, again, that the subjects are safe and the data is rigorous. Also, in the United States, we have the Code of Federal Regulations. I keep this on my phone. There's an app that you can download. There's a GCP app. It's not the most interesting, but from time to time, I need to go back and refer to it. The regs are written kind of gray, so sometimes it's hard to interpret, but it's really important to be familiar with those regs. The ICH GCP, there's several organizations, there's several, you'll see the Belmont Report, the Nuremberg Code, Declaration of Helsinki, the local IRBs, SOPs, sponsors, they all help develop the GCPs. NIH has also a policy, and in the policy it states that they should be well trained, your staff should be. They should follow GCP. You're responsible for the oversight of everything that's done in a clinical trial. And the principles of the GCPs help, again, for the safety and integrity and quality of the research trials. So when are they implemented? They're every part of the research study, designing it, conducting it, monitoring, auditing, recording, analyzing, and reporting data for clinical trials. Now, there's a handout and things where you can read this a little bit better. And I used to not read these, but I believe that if you live by these 13 principles, the ICH 13 principles, then you're doing your job. And if you start from day one, your life will be much easier than trying to go back and clean up later on. So what we're going to do is just kind of briefly kind of go through the 13 principles. I have these 13 principles posted in my office, all over the CRISP office. I hand them out. I constantly go back and review these because they touch base on every part of research. So clinical trials should be conducted in accordance with the ethical principles based on the Declaration of Helsinki, and are consistent with good clinical practice, applicable regulatory requirements. What does that say? It's saying that you need to follow these guidelines, and you need to make sure that everything that you do, that you're familiar with the guidelines and regulations, and that you follow them. Before the trial is initiated, foreseeable risk and inconveniences should be weighted against the anticipated benefit for the individual. Basically, it's going back to that safety issue. The trial should be initiated and continued only if the anticipated benefits justify the risk. I've had investigators look at me when they're consenting patients and say, I really don't need to go through the whole consent form. And no, the consent form process starts at the very beginning of the trial and goes throughout the duration of the trial. So every time you come in contact with that patient or subject, you need to review and make sure that they understand the risk and that they're well informed and that they still want to participate. Again, here we go with the rights and safety and well-being of the trial subjects. It's the most important consideration and prevail over all other interests in science and society. So I had a trial that I was participating in. We had about 40 patients enrolled, and we started getting safety reports. And the safety report said that this agent that we were using could cause strokes. So I went to the investigators and said, okay, so we really need to watch this. Well, about a week later, I had a subject that had a stroke. And honestly, we could not say that that stroke was not caused by the agent that she was taking, he or she was taking. So we started watching it, and it ended up, they had so many patients in the trial, and this is why you have data safety monitoring boards and stuff. They pulled the trial, and we had to bring in all the patients and do end of visits. We had to follow them and make sure it was safe. But this is why we do this. I mean, yes, the company probably didn't want to pull the trial, but it was the best thing for the patients. It's the safest thing. So it happens. We need to be make sure that we make sure it's safe for our subjects. The available non-clinical information on investigational products should be adequate and support the proposed clinical trial. The investigator brochure, our investigational drug brochure, is very important. It's kind of dry reading, but if you're doing a clinical trial that involves a drug or an agent or a device, you need to be familiar with all the things that have happened in the previous trials, phase 1s, phase 2s, and those so forth. Okay, where are we? Clinical trials should be, I think I said the available non-clinical information on investigational products should be adequate and support the proposed clinical trial. We did that one. Clinical trials should be scientifically sound and described in a clear, detailed protocol. The most poorly written protocols are the ones that have the most amendments, the most questions, and there's gray areas. And so if you're doing a multi-center trial, one area may interpret it this way and another area another way, and so you're not following to make sure it's generalizable and scientifically sound. So just keep that in mind.
So, you know, I had the opportunity to participate in a trial just recently, and when I was reading over their informed consent, you know, as a scientist, I was like, this is a lot.
It is a lot.
So my question to you is, when you're helping people develop these informed consents and these protocols, how do you weigh wanting to be brief and be able to be understood, but also being clear and making sure that you are describing the trial adequately? Like, where does that balance fall, and how do you do that?
So the question is, I'm supposed to repeat this, is when you're developing the informed consent, how do you balance and make sure the informed consent is understandable for the patients, because they are long and stuff like that. There's some key essential parts of the informed consent that you have to include. And with the common rule that just came out in January, what they're doing is they're doing a summary at the very beginning of the consent form. It's concise. It's supposed to be the key points. So it makes it easier for patients. When I consent patients, a lot of times you'll have a patient who says, it's a 26-page consent form. Oh, I really don't need to know this. But it's really important to go through the key facts. Also, my first trial, Dr. Ertaler was one of the investigators conducting the stress test. So what he taught me to do is go back to the patients and ask them, can you tell me what this trial is about? So after you explain it, just getting them to briefly tell you what you've explained to them. Because it does get long, it does get tedious. You can see them totally zone out. If you're doing an oncology study, those are really long, detailed consents. But the thing that's really important is, so in that concise summary that they're now requiring, is to make sure you hit those key points and make sure you drive. If this trial is going to cause strokes, if this trial is going to cause your liver enzymes to increase, or if you're doing a cholesterol lowering study, your muscles to ache, those patients need to be well informed of what those risks are and what the procedures are going to entail. We get into a lot, and we'll get into recruiting later on, but it's really important if the trial is going to make them come in every week or every month, that they know that up front and that you're recruiting the right patients for that. But each time when you're going through a 26-page consent form, you need to drive those key points. It wouldn't be a bad idea to have a checklist or something like that, just to go through those points with them to make sure they fully understand it. But getting them to come back and say what the trial is. My first patient, Dr. Erhard, was like, well, did she do the informed consent or perform the informed consent discussion? And the patient goes, yes. Well, what's the study about? Well, I don't know. And I was like, oh my goodness, you should know. So he kind of winked at me and he said, no, I can tell you it's about this and stuff like that. But having them, regurgitate, it does get tedious and it is long. And they're supposed to be written on a level that anybody could understand. And that's hard to do. So like medical terms, you need to explain. When you're doing biospecimens, and Dr. Edberg may touch base on this, explaining what it means when they say they're going to reproduce or they're going to store or they're going to use the samples for future use. Really driving in and saying, do you want to be informed about this? If they're doing genetic tests and they're going to give the results, are you going to give them the results? Do they want that? And keeping up with that is hard to do. And making sure that if they refuse, then they refuse. If they tell you no, that means no. Does that help answer your question? Okay. A trial should be conducted in compliance with the protocol that is received prior IRB approval. This happens all the time. Had an investigator on a Saturday call me and say, I found a patient for this trial. This was a real rare trial, but he didn't have IRB approval. And so you have to have IRB approval. We have investigators that a lot of times see, oh, my protocol's not really working that well, so we're going to change this inclusion criteria. We can enroll this patient and then I'll go get the IRB approval. No, it has to be approved before you do anything, even on amendments and stuff like that. The medical care given to medical decisions made on behalf of the subject should always be the responsibility of a qualified physician when appropriate, or a dentist. This is really important because a lot of times you'll let your research team write down AEs and SAEs, and then the research team is the one assessing. That's a big no-no. If you're the investigator, you should be making those informed decisions about your patient. You should say if it's related to the device or the study drug or the behavior or whatever you're studying. It's the PI's responsibility, not the study team. And a lot of times the study team, I have done some audits where the study team is assessing labs. That's not appropriate. So anytime you're making a medical decision on behalf of the subject, it should come from the investigator or a qualified staff person that has those credentials. Let's see. All clinical trial information should be recorded, handled, and stored in a way that allows it to be accurate, interpretive, and verified. I can't tell you how many times I've seen people put in their databases and not have source documents or not be able to tell you where they got the information. If you have an auditor that comes in, they're going to want to see exactly where you got that information. Whether it's from medical record or anything, you need to have a clear, clean trail. Also, if you make mistakes, which we all do, is just a single line initial and date. Again, single line initial and date. I have seen white out. I've seen blacked out things. I've seen scribbles. I've seen notes that are written out of order. These are all flags. These are all things that we're not following the GCP guidelines. Let's see. The confidentiality of the records that could identify subjects should be protected. This is a big one. If you tell in your informed consent that you are de-identifying the information, truly de-identify the information. Don't send, if you're doing a genetic study, don't send a blood tube or any information about that subject if you said that you're going to de-identify and they won't be able to identify who the subject is or link it to them. And I see this a lot of times. They'll write it all over the tube, and then in the informed consent it says that you'll be given a study number and stuff like that. So making sure you're following through the processes are really important. An investigational drug should be manufactured, handled, and stored in accordance with applicable good manufacturing practice. I love the research pharmacies here and at Children's Hospital. If you're doing a drug study, they keep logs, they make sure that it's under temperature, those kind of things. They also keep up with the supply, what's coming in, what's going out, make sure that it hasn't expired. If you're keeping it at your site under lock and key and hopefully under temperature control, you need to make sure you have those logs. I walked into an office one time and literally the drug was in boxes. Had not been logged, was not under a temperature. You know, in Alabama, one day it might be 110 and one day it might be 35. So if the drug says it needs to be maintained under these conditions, then please help make sure that it's maintained that way. And then systems with procedures to ensure that the quality of every aspect of the trial should be implemented. So having checklists, meeting with your staff regularly, talking about what's working, what's not working, talking about or going to a conference or the biweekly phone calls that some sponsors may have. It's really important to get the updates and make sure everybody's informed and that you have a systematic approach that is working. So those are in, the 13 principles are in your notebooks. I encourage you to just read them, even though they're really elementary, and I'm sorry I sat here and read 13 of them to you. They're really important. This is the outline of the ICH GCPs. It breaks it down in different areas. I included this just in case that you wanted to read a certain area. If you're more interested or your specialty lies into something like this, it starts off with the principles. It goes through the ethics committees, goes through the investigator responsibilities, all the way through randomization, progress reports, final reports, everything. It goes through sponsor responsibilities, and then it goes through also going multi-centered studies, record keeping, non-compliance, those kind of things. Clinical trial protocol and protocol amendments, it addresses that. It goes into safety, some of the quality control, QA, QC, and that's a big topic across campus right now, making sure you have a process that everybody on your team, you go back and you look at the information you're collecting and that it is systematic. We've had team members that have not kept records like they should. Let's just put it that way. They're messy. They're not organized. I walked in one office one time and there was papers everywhere. They weren't in a file. And what we discovered is participants were enrolled that had clearly met exclusion criteria that should have never been enrolled in the trial. And basically we had to start from scratch, putting together charts for everything. Regulatory documents should be filed in orderly too. So if the IRB came in and did an audit, they could find that you had approval and that you're up to date. Investigator brochures, you'll see multiple versions of this and making sure you keep the most current version and document that you've read and reviewed the versions. And essential documents for the conduct of the trial are there, and that you know you've done everything that you're supposed to do. So the difference in required regulations based on different types of research, really there is no difference. I mean, they all have their own regs, and here we break it down for drug studies and similar studies. They have similar guidance and regulatory documents. You have your 1572, you have those essential documents there. For medical devices, they're regulated by the FDA, they have letter of agreements, and they have a kind of a clearer process. It's kind of amazing. Of exactly what the documents that you have to do and what you, how you keep it and stuff like that. For any kind of natural history studies, you have regulations for that, and behavioral studies. We did a link here for you if you're doing behavioral studies, but really they all follow this 13 principles of GCP. Training in the basic GCPs, as you know, at UAB we have a GCP training. This is the link that gets you to the training. We expect you to do that pretty soon. And then you have the Miami CITP course, or CIT I course that you do basic IRB training for that. And this is all kind of looped together on the IRB website. You have competency domains, and in these competency domains, GCPs are incorporated in everything. The scientific concepts, the ethical considerations, medical development and regulations, the clinical trials operations, basically the implementation of everything, the study and site management, the data management, leadership and professionalism, and communication with teamwork. So here is a diagram of what we, if you're new to UAB or you have a new employee, this is kind of what we expect, and this is what we offer educational-wise. Within 1 to 2 weeks of employment, we hope that you have done the CITP, or CIT, I cannot say, I don't know why I keep putting GCP and that together, the GCP and HSP training. So go to the website, do those trainings. Within 2 months of employment, you go to the research orientation program. And this is more, it's geared more for clinical research, but we do include behavioral research and stuff like this. It's a very broad overview of research at UAB. Within 6 months of employment, we have the research training program and the CITP training program for PIs. That program is going to kick off April 9th, and we're in the process of updating and adding it, making it a little more basic at the very beginning, and then adding some other more advanced topics later on. The PI training program is a 4-week, every other week training program that investigators across campus come and talk, and you do some case studies and stuff like that. And then offered 2 times a month on the first and third Thursdays, we have the special topics in clinical research, are formerly known as the research seminars. Our next one is tomorrow here at noon, and we have someone talking, Dr. Carolyn Thomas-Jones, talking about the Diamond Project. Diamond is a grant that was written, and it's educational portfolio, research portfolio platform website. And so she's going to walk us through the use of Diamond and how we can also post trainings on Diamond. So it's a wonderful resource that we have that we don't tap into a lot. So she's going to talk about that. And then on March, I believe it is 7th, the director of the IRB, Adam McClintock, is going to come talk about the updates. He's going to introduce himself, make it kind of a meet and greet, and then he's going to talk about the Common Rule, the updates and things that are changing with the Common Rule. So that's what we have coming up. On April 9th, you'll see the CCTS Lunch and Learns. That's for the whole campus. We bring OSP, IRB, Research Administration, Encore, all these different departments come in and they give you the latest updates on research across the campus. Now we're going to move into ClinicalTrials.gov. And the goal for ClinicalTrials.gov is to improve the accountability and transparency of clinical trials. As you know, research, clinical trials is a public activity. So the public needs access to see what's going on out there. So ClinicalTrials.gov is basically a public access where they can see what all the different types of clinical trials that are going on across the country and internationally as well. We owe it to the people who volunteer for these trials to know what they're volunteering for and what the results of those trials are. So it guides research, it guides researchers in a systematic approach in putting their information about the clinical trials. Brief history, in 1997, there was a law mandating the registration of trials. Sadly, a lot of investigators kind of ignored this law and really didn't do it. In 2000, we had the implementation of CT.gov. So it was developed and it's publicly accessible online database operated by the National Library of Medicine. In 2007, the requirement for results from the FDAAA came out. And then from there, again, only less than 50% were registering and reporting the results of their trials. So there were serious discrepancies between what was reported versus the peer-reviewed journal articles, which shouldn't have been. And so the decision was made to re-implement the registration and reporting requirement, and harsh penalties were established. In 2016, the final rule was announced, and effective January 18, 2017, it went into effect. So everybody started scrambling around, making sure that they were registering their clinical trials. The NIH extended the requirements and announced grants that would be withheld for non-compliance. So can you see they're trying to get your attention to say, hey, if you don't register it, it's not good, you can't be published, you can't do this, you're going to be fined. Institutions are going to be held responsible, and individuals that don't do this are really going to be held responsible. So these are the required register reporting results. It breaks it down from the FDAA, the NIH, the International Committee Journal, Medical Journal Editors, and per Medicare. So it impacts all of this. Registration is expected, and then it breaks it down. Basically, for most of it, within 21 days of the first patient enrolled or subject enrolled. I have been made aware that investigators have not done this, and they were held, they couldn't publish results in journals. So it is real. They're really looking at when the patient is first enrolled, they do collect that information. Per ICMJE, it's before the first participant starts. So this is really important. At UAB, the IRB requires registration to be complete with NCT before the protocol is given final approval. Reporting results. So you register your trial. Now you have to keep reporting the results. This is the results is expected no later than 12 months after the last participant has completed therapy or treatment. This is really important. So if you finished your treatment and you finished you have 12 months to get that in. They do grant some extensions. We have staff that can help you with that, but it's really important to keep that in the back of your mind when you're making sure that you're keeping up with your trials and the results. Penalties. So these are the penalties. As you can see, it is quite extensive. They're trying to get your attention, and they will enforce it. Luckily, at UAB, knock on wood, we have not had any penalties. But if you do not follow these guidelines and regulations, we will. So I'll let you read through that. So in summary, there's 3 primary components to CT.gov. The registration part of it, the updates, so every 12 months you give updates are required, and then results reporting within 12 months of your last participant. The common rule, we talked about the changes to the informed consent. That's the most notable thing. But the common rule, there's some updates, and the IRB is still in process. If you go to their website, they have a really good training on the common rule and the updates to the consent form. They recorded it, so I encourage you to be familiar with those. And here are some accepted websites that can give you more information. So we at CRIS offer support, and we know it's tedious. We know it's overcoming, and they issue queries that a lot of times we don't understand what they're asking for. So we have a staff that provides support, and so we can help you with the registration. We can help you through the process of updating the website and also reporting your results. We do presentations for large groups, so if you have a group that doesn't really understand and you want to be trained, we can come to you and do that. We do regularly workshops about CT.gov to try to keep you up with the updates with that. And we help you when you have those queries. There's guidelines in answering those queries, and having so many days they flag. If you don't answer the queries and you ignore it, they flag it. And we're going to be contacting you because we keep up with those queries because we don't want UAB to be fined. So who to contact? These are the websites. Denise and Dorothy are excellent. They will come to you. They will try to make it easy for you. They're not going to do it for you, but they will guide you through the process. And these are some references. So study recruitment and retention. So we're going to totally switch gears and touch base on that. So why is recruitment important? And after you recruit, now what? So what do you do with this? And why is retention important? What are ways to recruit, and how can you improve recruitment? So successful recruitment is the first step to a successful trial. If you think about it, if you don't recruit and you don't follow the subjects all the way through, then what are you doing? Or why are you doing it? So, and if you don't screen the subjects, poorly screened subjects may result in a high dropout rate. And dropout rates don't help you get your results or see a complete picture. I was once in a trial that we discussed a participant. At this time, this participant was not homeless, but there were some red flags when we were discussing his recruitment or her recruitment. And we went ahead and we decided that it would be good because we could follow the subject and the subject would get more one-on-one care. But the subject later on in the trial, this was a 5-year study, became homeless. And so it was a huge challenge to follow the subject all the way through. But what we did as a group is anytime the subject would come in the hospital to be admitted, all the fellows knew my subject, and they would page me and say, hey, he's here, or she's here, you need to come follow them. So no, the information was not timely on those visits. They were out of window, so we had some deviations from the protocol, but we were able to follow that patient all 5 years and report results. It was a lot of hard work. So if you don't screen accordingly. And we saw some of the red flags, but we sat down with the investigator and said, what do you want me to do? You know, yes, it was a pain, but it was really important, and we got the outcomes. We've had patients that have been dislocated from storms and stuff like that. Those are extremely hard patients to follow, but we found them. They were located all over, but we were able to find them. So really making sure before you start a trial that you have the population. Really looking and doing the feasibility is really important. Not only does it affect your results, but it also budgetarily, it can affect a lot of different things. So know your population, know who you're recruiting, know what your study is asking. If they're asking for somebody to come monthly and they live 5 hours away, that may be hard. But if they live locally, that may be easier. If you're doing a pediatric study and they have extensive PKs and it's during the school year, missing school is hard. That's a red flag. So really knowing who you're recruiting in your population, and whether you have that population in the clinic, there are resources to help you form feasibility assessments. I2B2 is here at UAB, and then also at Children's, they have something very similar to I2B2. So running those reports and really looking, and then looking granularly at those reports. Do they live locally? What am I asking them to do is really important. Would you participate in the study or let a family member participate? This is something I ask myself a lot. And when you're designing studies, you need to think about this. What are they asking them to do? And what is the time that it takes? If you're doing an older population that is taking care, kind of that sandwich generation, taking care of their mom or their dad or aunt or uncle or something like that, it may be harder for them because they're taking them to doctor's appointments and stuff like that. So being flexible and making sure that maybe you can monopolize when they're coming to the clinic, that you can perform those study visits is really important. And how much will the study cost? This is something we don't often think about, and investigators are going to be held accountable for this. So making sure that you think through what you need to do that study and how much it's going to cost, and making sure you have the facility to perform the research study. So how will you contact your population? I need to add some things to here. Social media is a big thing. Facebook, Twitter, all of these platforms, some of them I'm not even familiar with, but there's wonderful platforms out there in social media that can help you recruit patients. Radio ads, television ads, the UAB Reporter are all good resources. I will caution you with radio ads and television is expensive. So when you're doing the budget, make sure you get those costs and you include that in your budget. I liked, and I don't know if they still do it, the online TKC message person. That was a free service. While they're sitting there waiting to make their appointments, you could play key points about your clinical trial. I hope one day that we have a kiosk in TKC or some of these clinics that they can go and look. On the CCTS website, there's a place that you can go to the website and patients can look and see all the different trials that are going on at UAB. So there's lots of different places. Mass mailing is good. I caution you, it depends on what kind of population. If you're looking for a specific disease, you may want to target those patients. But if you're, so if you send it out to everybody, you're going to have to weed through a lot of information to get to the patients that you can enroll. Community outreach programs, churches, health fairs, organizations, those are all good places. Senior citizen, organizations, those kind of thing, if you're recruiting that population. Patient referrals, that's another good one. Working with your colleagues. Hey, I have this trial going on. Going when you have faculty meetings and stuff like that and letting people know. I used to make a little card that I would give all my fellows to put in their lab pocket. I would regularly page them and say, hey, remember the blah, blah, blah trial. So flyers is another popular thing, brochures, cards, videos in waiting rooms, those are all resources that you can help in recruiting. Successful recruiting is reaching your recruitment goal. I always caution investigators when we're sitting down and doing that feasibility. If they say they have you know, 500 patients with this disease process, I'm like, okay, whittle that down. How many live locally? How many do you think realistically will say yes to this? I would much rather say a smaller number, hit that number, and then amend your contract and go above that number. I'm not saying that you can't go above it. Everybody wants to hit their goal and exceed, but being realistic in setting your goals. Staying within your budget, making sure you think through every process of that trial, you know, from your staff to the PI time to the resources that you're going to use, the tests, those kind of things. We're really focusing on budgets now, and there will be some workshops and some things. We use the Encore system, if y'all are familiar with Encore. We're going to start helping you put your budgets in Encore to be able to track that better. Making sure you're invoicing for things that if you're doing a drug study or a device trial and they're not paying you regularly, making sure it's written in your contract are important things to consider. And if you have an invoiceable, making sure that you go back and invoice. We leave thousands and thousands of dollars on the table that we don't invoice for. So coming up with that process, that thorough process, is really important. Retaining participants throughout the duration of the trial, I've kind of talked on that, but really you have to make the active role to maintain this. Get your staff, really relate to the patients, get them involved. So keeping them engaged throughout the entire study, setting expectations and communicating with them regularly is crucial. Using retention materials such as calendars, appointment cards, sending texts with their appointments is really helpful in getting them engaged. Sending them a simple birthday card or Christmas card goes miles. You don't even think about those little simple things, but they'll come back in clinic in January and go, I got your Christmas card. I really appreciated that. Or you wished me happy birthday. Thank you. So that personal touch, that personal contact, keeping them engaged with the updates that are going on with the trial is really important. They want to know when you get to the end of the trial, especially what group they were assigned to. So keeping them engaged throughout the whole thing and then letting them know, hey, you were in this group or you were randomized to that group, and continually thanking them for their participation. To contributing to research, science, and medicine. So sometimes you have to get creative. You know, my homeless patient, he went around 5 points. And so I was able to find out where he would visit and where he would work, and they would help me. I got his permission before, her permission before to do that, but being creative and doing that, going that extra step, really will help you in the long run. And then sitting down when you hit that brick wall and you can't think of, brainstorming barriers and helping to avoid them with your team, engaging your team is important. So if lost to follow up, re-engage. Just like the story I told you. It didn't matter that he didn't hit the visits perfectly, but we still were able to collect information. We were still able to say that, yes, they were alive. Have a patient contact list. I encourage you on the patient contacts list to regularly make it part of your visit to update this. People move all the time. Phones are disconnected. Getting contacts with people that don't live with them was vitally important for me to be able to follow up with them. So this homeless patient, I had his or her mother's information. I had a place that he would go regularly, or she would go regularly. So making sure you have those alternate contact numbers and that they're updated will help you in the long run. And making it easy for them to participate, provide parking. You know, at UAB, parking is a disaster. Making sure that you have transportation if you have them coming from the Webb building to CKC for a test, that you provide transportation. This happens, or Highlands. I mean, wherever you're doing it on campus, it's really important. If they're here for a long day, making sure that you provide meal tickets for them or even childcare. And retaining the participant throughout the entire study and informing them of the results. it's just something that we all need to do. Do y'all have any questions? Thank you. Oh, and I'm turning up the microphone.
Excellent. So I think one of the really important messages is to recognize that there's really skilled staff available to help you with every phase of this. So don't hesitate to contact the Clinical Research Support Office if you have any needs. You can access the contact information through the CCTS website, or just call PCAMs here and ask for help, and we'll get you in touch with the team. Thank you very much. We're going to take a couple of minute break. While you're taking your break, please take a moment to sign up for your IRB visit. So just to remind everybody, so we offer that 2 participants in this course every week can go sit in on an IRB meeting. The way it works, the IRB starts its meetings at 9 o'clock, I believe. So you will be released from, on the week that you've signed up, released from the class here at 9 to go join the IRB session. It runs till around noon. You need to be prepared to stay for the whole thing because we don't want to disrupt their activities. You're not meant to talk while you're in there, but you're there to observe. But it'll really give you a chance to see how the IRB operates and what the tone of a meeting is actually like, and you'll see what kind of issues they're wrestling with and what kind of things you can do in writing your IRB protocol to make it easy for them to do their job efficiently and quickly. So there's sign-up sheets on the table in the foyer here. So when you take a break, just sign up for your dates. We don't absolutely know whether the first date on the list is going to be available. So, but you know, if that's a good date for you, sign up. It's pretty likely it'll work out. If it won't, we'll reschedule you for a different date. If there are people off campus who want to do this, I don't think there's anything that precludes you coming in to do it. We just really need you to show up if you sign up for a date. You can call Jean Lambert to do that. If you have an IRB at your own institutions and you'd like us to help negotiate the right for you to go sit in on a meeting, we'll be glad to help you with that. So contact either me or Jean, and we'll work with your local IRB to try to help that to happen. Okay, great, and we'll reconvene at 9 for the second part. Thanks.
No, all that 15 people. Yeah, it's like,
Isn't it just
there's supposed to be one from each person.
ours were he put Sabuja and Samir up.
Well, I mean, here's the too.
I'm not sure your question got fully answered. How to balance the
My real question,
detail, but
yeah.
also.
My real question to her is I wanted to know, so what happened is I actually had my daughter participating in a study, and you know, they were consenting me as her mother, and the consent was crazy long. I mean, 15 or 18 pages. And I was like, I'm understanding what you're saying, right? But this study was enrolling a lot of people that would not have understood.
Right.
And, you know, I was like, is this really necessary to sit and go through all 18 pages when I think it really just confuses some people? And I think that the heart of her answer was, yeah, you have to do that. But really then at the end, coming back to that summary, because they did have a summary paragraph, coming back to that summary paragraph and making sure that that is what they kind of leave with in their head. Because I was like, you know, if I was just a normal, you know, Joe off the street, 18 pages in, I'd be like, I don't care anymore. You know, I don't even know if I want to participate. I don't know. And so that was kind of the heart of my question. She did answer that, but I just like, I was very taken aback by how long the informed consent was and how I felt unnecessary. But I think it is 100% necessary. you know, now.
It is necessary, but it's a real problem.
Yeah.
And the idea that we know what it means to write at an 8th grade level,
Mm-hmm.
if that's what the instructions say to do.
Yeah.
We don't know that, really.
Yeah.
And there are people that rewrite it, and that our staff are pretty helpful to make things comprehensible, but the whole concept of informed consent.
Because it's like informed, like, I put quotes around it now. Now that I've actually been consented, I don't put quotes whenever I say informed. Because, like, informed consent, you know, all the words are at an 8th grade level.
Right.
Whether or not that concept actually comes across at an 8th grade level is what gets lost. You know, the words are there, but the concept is not always there, I don't think.
Right.
So, I don't know.
Well, you know, it's the whole life experience goes into what helps you to decide whether something is something you really want to be part of or not. And
Yeah.
people, their experiences are so different. You can't meet everybody at the right place necessarily. It's no fun.
Yeah. I think so that.
But the concept of trying to get the person to explain back to you what they're doing and to ask them why they want to participate.
That is like, I'm going to recommend that to her. Because the girl that I participated, you know, I know her. And so I'm going to write her an email after this and tell her, like, hey, you know what you should start doing? Have them repeat it back to you. Because I think you'd be really surprised what they would repeat back to you about your study. What I would have repeated back would have been interesting.
So you remember the drill.
I was saying that also
training research.
So
very fast.
I try to run around.
Very fast.
Sure.
One review was like, see a little bit of data, I can see it might have been accepted.
Is this your last chapter?
The second chapter 2, chapter 3 is now just went online today. Yeah, but it's okay.
Mostly politically focused people, but it's a range, so this is alright. And it would be totally great if you would also tell them something about the sample processing core and stuff. So they know what some of the services are that the center has. How long? You have until 10 if you want it, but so how much it takes.
A couple presentations to do, but yeah. Do before that May 1st. Yeah, that sounds good.
Okay, we'll get started in just a second. I just want to remind people who are off-site to make contact with Jean if you want to do the IRB session. It's actually very strongly recommended. It's really a great opportunity here. So if you email Jean or call her, she'll arrange a time, she'll work with you to fill in one of the spaces that's available for the IRB training. So her phone number is at the bottom of every email she sends. So if you can't reach her right away, send her an email and she'll get back to you with a time when she can talk with you on the phone. Okay, so we'll go ahead and start the second part. So we appreciate Dr. Jeff Edberg joining us. He's the director of the Biorepository and sample processing parts of the CCTS. I guess it's supported beyond the CCTS, probably. He'll tell you a little bit about those aspects. His appointments in the Division of Clinical Immunology and Rheumatology in the Department of Medicine, and he's been organizing this for the UAB research community for quite a lot of years now, and he'll describe major issues around biospecimen, biorepository generation, and how they work.
Thank you. So please, let's keep this informal and ask questions, and if there's something that's obvious, we can move on and not obsess about it. And then probably the most important thing is that there are a lot of resources here at UAB already existing. There isn't a need to necessarily rebuild capacity in every division, department, building. There's a lot of ways to synergize. The CCTS can help a lot with your study, as you learned from Meredith's presentation with recruitment and regulatory assistance. We can help with the implementation in terms of the specimen management. A lot of specimen collection opportunities. There's a clinical research unit, which I'm sure you've heard about or will hear about. And we work very closely with all of those units to try to have a full service opportunity, so you don't really need to recreate everything for your own study. So as Meredith emphasized appropriately, in order to collect biospecimens for research, the first thing you got to do is have IRB approval. That's necessary and important in order to collect any specimen from participant, even if it's just blood from a quote healthy control, whatever a healthy control means. The CRIS group can help with language on that. We can help a little bit too. One thing I always recommend is when you're trying to collect biospecimens, don't limit yourself and be super specific in your consent forms. You know, don't say I'm going to collect a 5.5 mil green top, and it turns out those are not stocked anymore and you have to use an 8 mil. Well, you can't because you limited yourself in your consent form. So keep your consent form appropriate and honest to what you're doing, but don't get so super specific that you lock yourself into very specific collection types of tubes, or even the type of tube. Don't say a yellow top or a green top, just you're going to collect blood. You have to use volume, but you don't have to necessarily say what type of tube it's being collected in, because that can change a little bit with your study, and also what's in stock sometimes. Sometimes tubes get used up. If you're interested in biospecimen research and looking at something in blood, plasma, serum, whatever, oftentimes there are existing collections that are already available that you might be able to tap into. That certainly saves a lot of expense and time and can really facilitate your study. You can tap into something that already exists. A lot of times there are people that you know in your department or division that might be collecting similar samples that you can collaborate with. There are existing biobanks and biorepositories. At least at UAB, there isn't so much of a bank where you can just go and order something. We have a lot of investigator-initiated collections on campus, and people are very open to collaborating, but it's really in a collaborative sense, not in a service sense that there's a bank somewhere that just say, I need 52 plasmas from healthy males. But usually, if you approach me, I know kind of what people are doing in general and can kind of point to the right places to see where people are doing things. There are also commercial sources of biospecimens. That's always an option, but there's a lot of expense associated with that because it is expensive to collect, store, and make available. So that's kind of a last resort, I would think, just because of the expense. And you need to think through, if you're going to be collecting specimens, how are you going to do it? Is it going to be done during a routine clinical encounter or is it research-only encounter? And the only issue here really is if it's during a routine encounter, you just need to think through how those extra tubes and where those extra tubes are going to get drawn. Typically, in our work, I work primarily in lupus, we try to work with the physicians that are seeing the patients and give them the tubes that we need to take with them to TKC. And the TKC lab is very good about drawing extra tubes with the appropriate form. And there's a little bin that they put it in afterwards that the coordinator can pick it up. Just need to work that through with whoever's seeing the patients to make sure that consenting occurs before they go to the lab, that the tubes are available so when the patient goes back to the lab, they can give with a bag of tubes and ask them to please draw those in addition to whatever tubes are going to go off their routine encounter. Usually there aren't charges associated with that. It really depends on scale. Maybe doing this on thousands of people, the lab would probably start to say, you know, what's going on here? This is becoming a little obnoxious. But a couple tubes every now and then, the lab is usually more than happy to help out with. And actually, I think they're also over in West Pavilion on the second floor where pathology is, the outreach lab. I believe they have a phlebotomist on duty during, I think, normal business hours, not after hours. There's a charge for that, but you can't have people go over there to have blood drawn. That's a need too. Sorry. So if you decide that you really need to build a collection of specimens, you really need to think through, so what is a biobank or biorepository? It's really your library of specimens that are stored for your use. Typically, specimens are going to be, when they're stored in a laboratory, are not stored for clinical use. There's a lot of issues with clinical use of specimens, chain of custody, and appropriate labeling and things like that, that usually preclude most research laboratories from having clinical samples in the lab. There are some exceptions that we actually have one study where we are storing tubes that are being used for clinical sequencing on individuals who are suspected of having inherited genetic diseases. So there are ways to work around that, but you really need to work in the context of a lab that has appropriate certifications to handle those kind of samples. Typically for research purposes, it doesn't mean you need to be less careful. It just means that there aren't as many regulatory requirements that you have to follow, but you still need to follow good laboratory practice. And again, just to reiterate, the specimens you're going to be testing or want them to be tested for any clinically actionable finding or something that's going to go to that medical record. There's a separate path that those samples need to go through. They don't go to your lab, get divvied up, and then a little bit goes over to the clinical lab for testing. You have to have a dedicated tube to take care of that. And staff who work with samples that are going in the clinical pipeline need to have appropriate certification, typically CLIA or CAP, College of American Pathologists certification. The lab needs to be certified to function in that type of setting. So one of the most important things, and it sounds kind of silly, but you'd be surprised at what we see, make sure your samples are labeled. I've literally seen samples, vacuum containers thrown into styrofoam cups, thrown into a freezer, where someone scribbled something on there that I guess is text, but you really can't tell. You really need to make sure that your sample's labeled, that you know what it is. And at a minimum, you need an ID or something to identify that sample. You need to have data associated with that sample too. So I've got a sample in the freezer from a patient. Well, so what? Who is that patient and what kind of data are associated with that patient? So if you're running a study, you've got to make sure that you got some sort of way of tracking, presumably basic demographic data, any phenotypic data that you think might be important for your study, and then any research-based results. And of course, all this would need to be disclosed in your consent form if you're going to be collecting personal information. Typically, in our studies, in our consent form, we always say that we will review your medical record for study-related purposes. That allows us then to derive information from the medical record to keep as part of our research results. You need to think about what samples you're going to bank. So not all specimens are good for all assays, right? So there's plasma and serum. They're not quite the same, right? Serum is clotted. Plasma has an anticoagulant, so it doesn't clot. Some anticoagulants like EDTA, for example, can interfere with a lot of different assays. So you need to think through what it is you're going to do with your samples so you can know what kind of blood to collect. We get questions all the time, well, what should I collect? Well, I don't know. It depends on what you want to measure. That dictates what your collection procedures should be. You need to think about the phenotype. So a lot of times people say, I just need a healthy control. Well, what's healthy? I mean, is anyone really healthy? I work in lupus, so we're interested in systemic autoimmune disease. So for me, we typically don't screen for hypertension or cardiovascular disease. We're really interested in systemic autoimmune disease like arthritis, lupus, vasculitis. So our healthy population may not be appropriate for other individuals who need, quote, healthy individuals. So you need to really think through what's your phenotype and what are you trying to do. There are a lot of issues of the type of specimen tissue versus liquid, blood, urine, CSF, saliva. Tissue is much more difficult from my perspective to deal with. There are labs in pathology that are very good at handling tissue, both in terms of collection, sectioning, embedding, and storing. And I'll have some links to that in a few minutes. There's a lot of interest in studying the microbiome now too, which requires collection stool. There are resources at UAB also that will help with collection, DNA isolation, and sequencing for microbiome analysis. So a lot of available tools. At the end of the slideshow, I think there's some web links that you can access to find out how to get more information about those. Now here's one. So our lab deals primarily with so-called liquid specimens, blood, urine, CSF, and you're welcome to contact me, or there's the lab number at the end of the slideshow too. We can help if you just want to talk through what you're doing. That's fine. You don't need to use us if you have your own lab and want to do your own thing. That's perfectly fine. We're here to help. We are here to work with studies too. So if you do need help doing things, we are available. Pathology has a great tissue procurement facility that's run by Dr. Grizzle. Dr. Grizzle is a national expert in tissue banking for research. And he runs a facility over in the Ziegler building. And I'd strongly urge that if you think you need tissue for your research, that you contact Dr. Grizzle or Kathy Sexton to talk through what it is you need to do. They do have a limited number of specimens that are available in cryogenic storage, but typically, if you have a need, they'll kind of assess what it is you need and then kind of develop a schedule on how they might be able to derive those tissues more in real time through routine clinical care. And then more recently, AP in pathology has expressed an interest in making sections available for research that they're using for routine clinical use. So once a section is assessed, there's a lot of leftover material that currently just sits somewhere on a shelf and doesn't really get used. So we're working with them and with informatics and CCTS to try to come up with a way of inventorying those and making those specimens discoverable in the sense that you would be able to find out that there's a section of whatever organ you might be interested in that you can then work with the staff of anatomic pathology to find out about how to get access to that material for research. So if before you start collecting samples, you really need to think through a number of different things. As I mentioned, you got to think through the type of material that you're going to be collecting or drawing. You need to think through how it's going to be handled. Some assays that you might do might have very sensitive analytes that are sensitive to temperature and time. Those are usually the two most important issues. You need to know that ahead of time. You need to know how the samples can be stored. Do they need to be -80? Do they do cryogenic storage? Is -20 okay? Then you think about, if you're going to put them in a freezer, how are you going to find them after you put them in there? Most laboratory freezers are probably not the most organized environments. And you just need to think that through. Our freezers in the CCTS, they're all fully inventoried down to the tube level, so we know where every aliquot is, but we've got specialized software to do that. And it takes a lot of effort and a lot of time to manage that. I'm not sure that every lab needs to do that necessarily, but you do need to think through to make sure your freezer is organized so you can actually retrieve the samples when you need them. And you also want to think about potential future use of samples. So we do a lot of work in genetics of lupus, which means we need DNA. But if I get a purple top tube, I can get DNA from the white cells, but there's a lot of plasma there. So I always save that plasma. A lot of it doesn't get used, but oftentimes it does. You never quite know what's going to happen in the future and what your needs are going to be and what questions might be important, or whether there might be collaborations. A lot of our samples have been used in collaborations around the world where we can become part of other studies because we've got the specimens to participate. So is the way we kind of think about specimens is kind of this life cycle, going from specimen collection to ultimately to specimen use. And at each step in the process, there are issues you've got to think through and understand what the pitfalls are. Obviously, specimen collection, you've got to understand what it is you're collecting and make sure you're collecting the right material. When you're collecting specimens, you're going to be testing the samples. You need to disclose and inform consent. Will research findings be shared with the participants? Typically, that's no, but sometimes it's not. It just depends on what your study is. Need to think through incidental findings of potential clinical importance. So we found lupus patients once that turned out to have Klinefelters, but it wasn't tested in a clinical lab, so we really couldn't report that directly to the patient. We had to go back to the referring physician that sent us that patient referral and have that physician work through with the patient to follow up on the finding. One other thing about future studies, when you word your consent form, don't be close-ended. Just don't say, I'm going to measure X, Y, Z, and then we're done. We always try to say, we'll measure specialized laboratory tests as the research evolves. So we don't limit ourselves to just measuring a few things that we know we're going to measure right away. Because again, you don't know what's going to come up a year or two down the road. In your samples, there have been a lot of changes in the so-called common rule, which I'm sure you've heard about, or if you haven't, you will soon, which are the rules and laws that regulate informed consent. There are a lot of changes in how biospecimens are handled from a common rule perspective. Some of the issues have become a little bit easier now. It used to be that if you had an IRB protocol for just the analysis of samples that you've already got, you had to do a yearly renewal. That's now going away. You don't have to do a yearly renewal of that. There are a lot of issues about what you can disclose. And these are issues that if you're going to be collecting specimens, you really need to understand. You can work with Meredith's group to make sure that your consent form is worded appropriately so that you're in compliance with the new guidelines. So just a couple words about the different types of samples. So this is going to focus primarily on blood because that's what we mostly work with. Remember, plasma serum is the liquid part. Plasma is anticoagulated, so I usually have EDTA, citrate, heparin to prevent clotting serum. The blood is allowed to clot before centrifugation. They're very similar but not the same. Again, you just need to understand what it is you're measuring and know whether plasma or serum is appropriate. You can also get nucleic acids from cells in blood, typically DNA and RNA. There can be, from a buffy coat, which is the white cell layer of blood, you can derive either DNA, RNA, or you can do both. They use different extraction procedures, but it is possible to do both. There's also the emerging concept now of circulating DNA and sometimes RNA, which some people have conceptualized as a biopsy, a liquid biopsy where you draw blood and look at DNA in the circulation, sequence it, and there are, one can look particularly in malignancy and cancer, you can find characteristic sequences that are associated with tumors that can be used diagnostically for early diagnosis. That's still in its infancy, but there's increasing interest in measuring circulating DNA and sequencing it. There are specialized collection tubes because circulating DNA is very susceptible to degradation. It's very low concentration, so it's easy to lose. So there are some specialized tubes that help preserve the circulating DNA. And then certainly cells, you can get white cells from blood, and those can be isolated out through centrifugation techniques. They can either be used immediately fresh or they can be cryopreserved. Our lab can do that, or we can tell you how to do it if you want to do it yourself. It's not particularly difficult, just requires a little bit of practice. So as I mentioned, there are a lot of different types of blood tubes. Becton Dickinson has a chart that you can get at that website that tells you all the different color tops and what's in them. Most of these are anticoagulated except for the red or so-called tiger top or golds. Those are the serum tubes, those clot. The one thing with serum tubes is oftentimes they come with a little wax gel barrier in there that helps separate the serum from the clot. It's really good for isolating, maximizing yield of serum, but sometimes there's a clot activator in there and that can interfere with some assays. So again, you just have to know what it is you're trying to measure and just make sure that the tube you're using is compatible with what you want to do. A lot of different kinds of anticoagulated tubes. It really depends on what it is you're doing on the sample. The most common ones that we use are the green and purple and yellow. Green being heparin, purple and lavender EDTA, and yellow ACD. There are some differences. I'll get into that particularly with DNA. So if you're trying to get cells, obviously you don't want your blood to clot because once it clots, those cells are going to be all messed up in that and then fibrous clot. So you need to use anticoagulated blood. The anticoagulant can make a big difference. So in this case, particularly EDTA is really bad for B cells. They don't tend to be viable after isolation. So if you're interested in B cells, don't do an EDTA tube. We usually use heparin green. Yellows work fine too, but not EDTA. Time and temperature is important for cells. Particularly neutrophils are very short-lived, and you just look at them the wrong way, they become activated. So you need to be very sensitive to what cell type you're interested in and how you're going to handle your blood. You can't collect blood and let it sit for a couple hours. You're going to do neutrophils. You really need to get going right away. If you're doing PBMCs, peripheral blood mononuclear cells, and time is important, BD cells a CPT tube that actually has a matrix in it that allows you to draw the blood directly in the tube and just throw it in a centrifuge, and it will separate out the PBMCs from the red cells and the neutrophils. Typically, those work pretty well. They're more expensive than doing it by hand, but you have the convenience factor and the speed that you just throw it in a centrifuge immediately upon drawing. So I've already mentioned plasma serum. Again, time, temperature, always an important set of variables. Some analytes are very sensitive to handling conditions. They need to be stored right away. Some might be light sensitive too. I always get confused. Some people think vitamin D is light sensitive. Some people think it's not. But you just need to be aware of whether there's light sensitivity. There are amber colored storage tubes that you can use if you think you have something that's light sensitive. So here's a serum collection tubes, and in the picture you can see the tube on the top there has that little yellow gel material at the bottom. That's that gel matrix. That's really good for maximizing your serum yield, but just need to make sure that it won't interfere with what it is you're measuring. Sometimes people call these tiger tops. They're kind of this reddish color with the black striping in it, kind of shorthand. And these are the typical plasma tubes that we use with anticoagulants, heparin, EDTA, or ACD. There are some plasma separator tubes, which also have a gel matrix in it. Most of those, I think, are heparinized. Again, it helps maximize yield of plasma, and they're very easy to use. They're more expensive, and you need to make sure that the additive that's in it will not interfere with your assay. So a few words about DNA and RNA. So typically, DNA, when you're going to use DNA or RNA, you're going to do PCR reactions at some point in your workflow. And heparin is a great inhibitor of Taq polymerase. So typically, you want to avoid green top heparinized tubes when you know you're isolating DNAs. We recommend purple. That works very well. Yellow tops can work too. Typically, from blood, you can get enough DNA to do just about anything you want to do from a single tube of blood. It doesn't really matter the volume for 8-10 mils. There's a lot of DNA in blood. Most modern genetic work requires very small amounts of DNA. But if you have a rare patient population or your access to patients is limited, you want to make sure that you do get enough DNA that you can stock it away and not run out, because that technology is constantly changing, and every couple of months there's a new way of analyzing DNA, new sequencing techniques. So you want to make sure that if you're putting all the time and energy into collecting and phenotyping, save some and make sure you've got enough that you can go back and do additional studies. I already mentioned the cell-free DNA or RNA. Those can have potentially diagnostic benefits, though that's still kind of an active area of research. RNA is very sensitive to handling. There are RNases which degrade RNA everywhere. And so if you're going to be isolating RNA, you need to make sure that you're using an RNase-free workspace. There actually are specialized tubes called PaxGene tubes that are used that have a stabilizer in it. So it actually lyses the blood and all the cells and stabilizes the RNA. And those can actually be stored in the freezer for years after collection with little degradation. You can never say no degradation, but little degradation. There are also reagents you can buy that if you have already drawn blood that you can add to the blood. One is called RNA later. There are others too. But again, it's the same idea. It lyses the cells and stabilizes the RNA and inactivates the RNases. Where RNA comes from is actually very important. If you think of blood, blood is a very complex tissue, right? A lot of different kind of cells in blood. And even when you talk about T cells or B cells, there are lots of different subsets of T cells and B cells that all have different functional properties, and they have different transcriptomes, different repertoires of RNAs encoding proteins. So if you're getting RNA from blood and you're going to be doing an RNA-seq experiment and looking at the transcriptome, that's really going to be, it's going to be largely a reflection of what the composition of your blood was, what the white cells were. So if you know you're interested in a particular type of T cell, oftentimes it's better to isolate that T cell out first and then extract the RNA. That is possible, but again, time is an important variable because RNA will start to degrade even in intact cells once they're going outside of the body. But just keep in mind that if you're doing RNA analysis and doing whole blood, you've got to think through the compositional issue. And particularly when you go to disease populations, we're interested in lupus, white cell counts change dramatically sometimes, both as a function of disease and of treatment. And so you need to be aware of what types of cells are in your mixture. So now some of the practical issues, once you collect samples, as I mentioned earlier, you gotta make sure they're labeled so you know what they are, and it has to be legible. And don't use markers that will smear, because oftentimes these get put in a refrigerator, they put in an ice bucket, and they get wet. And if you're using a marker that smears, then you can't read what you got. So really think through how you're gonna label your samples. Our lab uses all pre-printed labels. I know not everyone can do that, but if you're using our lab, everything will be labeled with a pre-printed label that won't smear or come off. And you think about specimen transport, so there's a logistics of your patient being seen at TKC or WIC or wherever. Blood is drawn there. How does it get to where it's going in a timely manner? Does it need to be cold or not? Those logistics oftentimes are very difficult to work through. We don't have staff to be running around collecting samples. We tried that once and we ended up not being able to do any work because every time we got back, someone else had something else to pick up. That's just a non-starter. As a study, you really got to think through how you're going to get the samples from where they're being collected to where they need to get. And then you need to think through the processing procedures on how the sample is going to be worked up. And then storage obviously is important because typically you're not going to be analyzing the samples right away. You may wait until you get a certain number to do an assay. So you got to think through how they're going to be stored, and does storing affect whatever it is you're going to measure. One of the big issues is freeze thaws, particularly plasma serum. And it's important to track that, to know if you're thawing the sample. Sometimes people just put a little mark on the tube with a Sharpie just to indicate that it's been thawed. In our software system, we actually can track freeze thaws. Every time we take a sample out, we indicate that it's been thawed and when it was thawed, so we have a record of that. But that's important because if you have an analyte that really needs to be used on a fresh or not previously thawed sample, you need to get a fresh sample or one that's never been taken out before to analyze. And as I mentioned before, tracking where the samples are in your freezer or wherever it is you're storing it, you just need to know where your samples are. That sounds kind of silly, but you really need to think that through. So where can deviations occur? So a lot of times people collect the wrong tube, or, well, I ran out of green tubes, I'll get a purple top. Well, maybe, but you need to think that through and make sure that it's going to be compatible with what it is you're doing. Here's some examples. I guess it doesn't show very well, but you can see that the label on that tube on the left is almost ripped off. You need to make sure the tubes are labeled so you can read them. This transport issue, so how the samples going to get further drawn to where they need to go. We work with a lot of clinical trials that are occurring all over campus, and we're constantly running into issues of people get busy, they forget to pick them up, or they can't pick them up because they're doing something else, they're picking up a different sample. Just need to make sure that you've got plans in place to make sure samples get to where they're supposed to go in a timely manner. And again, the issue of, are they handled properly? Are they light sensitive? Do they have temperature requirements? Do they have to be refrigerated or not? Those are important points to think through. Processing of samples is important, obviously, too. You need to make sure that your samples are processed in a timely manner. Again, that's really dictated by what it is you want to do. If you're getting DNA, you know, those can sit actually for a couple days in a vacuum container, even at room temperature, and you can still get good quality DNA for analysis. If it's RNA, you can't do that. So you just need to understand what it is you're doing and what the parameters are for time and temperature. And if they need to be centrifuged, you need to make sure that they're centrifuged the right way. 4 degrees, what temperature, what speed, all those kinds of issues. Make sure your samples are stored correctly. So as I mentioned, everything we have is in boxes and inventory down at the individual aliquot level. Just need to think that through. And again, that freeze-thaw cycle is important. And again, hopefully this is common sense, but your results are only as good as the conditions of the biospecimen. So you put work in up front, make sure you collect the right samples, process them correctly, store them correctly, then you'll have interpretable results. And again, we're here to help. We don't have to do it for you. We can if you want, but we're here to help. And if you've got questions, we're happy to talk things through. So the most important thing that we find, particularly in clinical trials, is oftentimes the study coordinators don't really understand their protocol. So I can't, again, it's common sense. Know your protocol. Know what's supposed to happen. You know why you're doing things. Know when the participant's supposed to come in and have samples collected, particularly in the context of a trial. Know where the samples are going and know what's supposed to happen to them. And make sure you work with your coordinators and staff, if you're not doing it yourself, that they understand what's happening. It's a lot of times I find that coordinators are intimidated by lab stuff because I think it's complicated. This stuff really isn't complicated. It's really very straightforward, common sense, and we're here to help, and we can help them understand what's happening so they can have a better understanding of their own protocol. So I mentioned centrifugation. This causes a lot of confusion. So the speed of centrifugation can be really important for some samples, less so for plasma and serum, more so for cells. And there are really 3 variables. There's time, how long do you spin it for, how fast do you spin it for, and what temperature do you spin it at. So 4 degrees, you're doing neutrophils, that's really bad because they're going to clump up and you're done. You're not going to do anything with it. So you got to know what the appropriate variables are for your sample. And the issue with centrifugation speed, people always get confused, is you have RPMs, or revolutions per minute, which is pretty straightforward to understand, but the diameter of the rotor will change the force that is being exerted on the sample. So then you have G-force, and G-force is an absolute. Every centrifuge can spin at a particular G-force, but the RPMs that required to achieve that G-force will be different depending on the radius. So you just need to be aware that there's a difference between times G and RPM. They're not the same. And we've actually had people walk in the lab and say, well, here's my blood. Well, what do you want us to do? Well, just do the usual. Well, there is no usual. You got to think through these things and know what's supposed to happen. Again, labeling, we do pre-printed labels that are generated through our inventory system. And actually, we use this system called Encore that Meredith mentioned in the previous talk, this clinical trials management system. It has a biospecimen component to it that we use. So if you're actually running a clinical trial, your samples are associated with that same trial, with the same patient records that are already in Encore. But that system allows us to pre-print labels that are barcoded, and we use a particular type of label that it will not come off at cryogenic temperatures or -80 storage, and won't smear. So barcoding is something that not everybody has to do, but it does allow for every aliquot to have a unique identifier. Though generating barcodes requires specialized software, that's something we can help you with if that's important to you. The problem with barcodes, if you have barcoded sample, you have a barcode reader. Most labs don't have barcode readers, so it doesn't do a lot of good to barcode if you don't have the infrastructure to really take advantage of that. You need to think through the type of label you're using. So if you're going to a cryogenic freezer, that's -150 to -180 C. Not all labels are compatible with that type of low storage. So you need to make sure that the label you're using is appropriate, and we can provide guidance on the labels if you need help with that. And one important thing we always face, we get a sample, well, I wasn't able to get the ID in time, so just work it up, we'll give it to you later. Well, we can't do that, because once the sample's in the freezer, you really can't relabel it, right? Because it gets frost, it gets wet, and labels are adhesive, and they don't stick when it's wet. So we've got to get those labels on the tube before they're put in the freezer. That's just really important. I mean, you can try to relabel, but they typically come off after 1 or 2 uses. 3 different types of freezers. There's kind of the -20 freezer. That's like your household freezer temperature. But as you know, most household freezers don't freeze up. They have the defrost cycle, which means that they're cycling the temperature. That's not good for specimens. So if you're going to use a -20 freezer, make sure it's a non-cycling freezer. That's the type that builds up ice. It's a real pain in the butt because you have to defrost it every now and then, but your samples are safe that way. The better way is to use an ultra-low freezer, -70, -80 freezer for long-term storage. And then there are cryogenic freezers, which typically are used for storing cells. This is based on using liquid nitrogen. Those are held at typically -150 to 180, -180 degrees C. There are mechanical freezers, compressor-based, that can go down that low, but they're really expensive. They're very high maintenance, and liquid nitrogen is a much easier way of doing that. Think through how you're going to store your samples. Everything we do are in racks and boxes. We don't throw them into the shelf or have these kind of open racks like this, or styrofoam cups is my favorite example. Those are not good ways of doing it. And we can, if you want to try to do something like this, we can help with what we buy. And when you think about freezers, you need to really think through, do you really need another freezer, or are there other options on campus? We've got, not unlimited, but we do have a fairly extensive capacity over in the Shelby building. And we actually do work with a number of studies where we store all their samples in our freezers for them. It's not free. I mean, we have to cover our costs, but that is an option. And just need to think through, freezers are very expensive. You have to maintain it. And if you really don't need a whole freezer, you just need a couple boxes or a rack or something. Can you work with a colleague, or you might want to talk to us and see whether that might be feasible to store in our facility. There are a lot of different kinds of ultra-low freezers. The last couple of years has seen the release of a number of new energy-efficient freezers, actually. The -80 freezers use an enormous amount of electricity. The Shelby building apparently is the worst building on campus for electrical usage, primarily because of -80 freezers. And it's a compounding problem because when you have a -80 freezer, it generates a lot of heat, which means you have to have more AC handling capacity, which leads to more energy usage. The newest generation of freezers have a much lower energy consumption and lower heat output. And so we've started buying only those types of freezers now. There are a couple different kinds. There's actually one that doesn't need to use a compressor. It's something called a Stirling engine. That's the type of freezer that's used on the space shuttle, actually. It's an interesting technology. We've actually had some problems with some of ours, so we'll see how that works. Traditional compressor-based ones are the ones that are most commonly used, but the newer generation compressors are much more energy efficient. They're also much quieter. Most of you probably know -80 freezers can be really loud. You don't need to near them. The new ones are really, really quiet. So you just need to kind of think that through too. The newer ones typically are more expensive in the short term, it's the upfront cost, but the long-term costs are much lower because you have lower electrical usage and lower AC requirements. So it is important to maintain freezers. Most labs don't do this, but you need to watch. There are trends. When a freezer starts to go down, you can actually see that ahead of time, usually. We maintain all of our freezers with daily visual inspections. We've got 2 redundant monitoring systems, one of which is 24/7 online that we can just log in anytime and see what's going on. The temperature. The second system is hooked up to the institutional system called CAMS, and there's a call list that if anything happens and an alarm is generated, they call us and we can decide what to do. Make sure your staff is obviously trained both in knowing how to use the freezer and how to spot when there are problems, and regulatory training. Bloodborne pathogen, that's another one that's important, I forgot to mention. If you're handling biospecimens, you need to make sure you do bloodborne pathogen training. That's the first thing to do. It's online on the UAB learning system. Just a few words about specimen tracking and management. We use the system called Encore, as I mentioned. And what that does is allow us to track every specimen within the freezer, and all the times that we intervene with the sample or touch the sample, we can monitor or record when we do that and what we do. So it's an audit trail that shows who made the change and what the change was. It can track the full specimen life cycle, so we know when it came into the lab, we know when it got stored, we know when it left the lab, when it came back. For our purposes, we have about 250 studies on campus we work with, so obviously reporting functions are important so we can know what we have and report out to people what we have for them. And we can import bulk specimen collections. We do that quite a bit now. So if you wanted to sort your samples with us, we would ask for a spreadsheet of what it is you have, and then we would import that into the system. It's just a screenshot of Encore. And the point here is that this is at a patient level, so every patient will have a record, and then there are what are called cases. That's every encounter. So every time a patient comes in for a visit, there's a case generated, they generate the case for all the specimens. And then associated with these specimens are all the detailed information about times, dates, what it is, how much it is, where it's located, and then other information might be important. So for DNA, for example, we keep track of concentration. OD26280 is a QC measure, where the DNA came from. Did it come from blood? Did it come from the cell line? All that can be associated with the sample. And this is just a storage view, just showing, so if you think of a freezer storage box, you have those little grids in there, and we just number those from 1 to 81 or 1 to 100, the grid. And this will just show you that every grid level has a sample associated with it, so we can know exactly where every aliquot is of every specimen. There are options for individual labs to do this if you want. FreezerWorks is one that we used to use. That's probably the best one. There are other systems too. FreezerPro, OpenSpecimen. There are laboratory information management systems. Those are typically very complex and very expensive. Those are typically used by core labs that are running large instruments that generate huge amounts of data. Typically, individual labs aren't using LIMS. But those are options that you can run on your own computer. You don't need to be centralized on a server. Typically, options that are not good, keeping paper records, they tend to get lost or destroyed. Excel spreadsheets, you know, the problem with Excel spreadsheets is people try to get fancy and sort, and they forget to sort the whole thing, and then everything gets jumbled. You don't know what you have anymore. So I would recommend using a database like Access, which most people have, if you really want to keep records yourself. If you can't use one of the inventory software programs. But just be careful in Excel. I mean, Excel is fine, it's a great program, but it's the sorting that usually gets people messed up if you don't need to sort the entire table. So now, the last thing I'll talk about is associating data with your specimen records. And you know, certainly you're going to be running, when you're collecting samples, you're going to be running assays, tests, whatever, whether it's sequencing or assays on plasma serum, you're going to want to keep track of that. But you also want to keep track of dates and times when the specimen was collected, potentially even who worked on it, so you know if something is funny or goes wrong, you know who to talk to. The issue of clinical data association, again, you have to make sure that you do this in your informed consent, that you're going to be associating clinical data with your research-based specimen. That's fine to do as long as you disclose it. There are a lot of different ways to do that. Oftentimes people use you know, simple data collection tools like the design, case report forms. But more recently, there's now the ability to link directly to the EMR and derive data from the electronic medical record. That, in many ways, is the best way, though there are some complexities associated with that. As you guys know, the EMR is not a panacea of giving the answer to everything you need. If it's not codified in a way that is useful to your research study, then it's not going to be available to the EMR. So for example, my case in lupus, a lot of times, so in order to have lupus, you have to have American College of Rheumatology criteria. Those are not documented directly in the medical record in discrete fields. It's usually in the comments, there's a lab test, and so yeah, it's there, but it's not something that's easily extractable. But lab results are more easily extractable. So you just kind of think through, if you want to link to the electronic medical record, what kind of data are you getting out of it? Meredith mentioned I2B2, that's informatics for integrating biology in the bedside. This is a great system that we have here, and what it does is it actually has an electronic data warehouse that takes a dump from the live medical record system every night and puts it in a separate place, so you're not actually querying the medical record system. It's done safely somewhere else. And you can actually query, I mean, the first level query is just kind of an anonymous cohort identification. How many patients with lupus are there at UAB? You can get aggregate number 552, whatever the number is. if you have IRB approval, you can actually drill down and actually get information, identifying information about those patients and clinical information. What we're doing is we're actually integrating Encore into the ITB2 environment so that you could actually run a search given diagnostic or laboratory-based criteria. Is there a DNA sample associated with somebody that might have those criteria? And that's live now. You can't find out who it is or who has the sample. Typically, these are investigator-initiated studies that have collected those samples, but it will tell you that it exists. So in this case, they just did SLE with DNA, and it says there are 36. That's knowable who they are, but you at the front end can't see that. You'd have to get with the ITB2 team to say, hey, I'm interested in following up on this. If you have IRB approval, then they'll put you in contact with me and we'll figure out who has those samples. And again, that will be in a collaborative sense. It's not a bank where you can just go in and say, I want those. You can try to work with the investigator that collected the samples, see if they are willing to collaborate with you. So how can you get more information? So we're usually here the first Wednesday of every month in PCAMs from 11:30 to 1. That's just a walk-in clinic. Anybody's welcome to come. Free lunch, if you want free lunch. And we're happy to talk through any questions about study design or study implementation that you might have. And here's some contact information for me and for the labs. We actually have 2 separate labs. As I mentioned, the main lab is in Shelby. That's where most of our freezer storage is. We also have a lab up in Jefferson Tower that's physically located within the clinical research unit in the phase 1 unit on the 15th floor. That lab primarily works with specimens derived in clinical trials, but we do other stuff there too. And then some web links for other information. There's OHS, IATA, so specimen shipping I didn't mention, but if you're going to ship specimens, you're supposed to have actual certification to ship because they're potentially infectious materials, so they're considered diagnostic specimens, and you need to be certified in order to legally ship those. ISBR is the International Society for Biological Environmental Repositories. There are a lot of good best practices on specimen handling and storage there, and another website for specimen handling. Okay. Any questions?
So you mentioned several times software that you use to follow stuff. So now that most data are kept in the cloud, is the issue of software becoming outdated and being unable to be updated sort of gone away, or is this still a major issue that we all have to deal with all the time? And how do you
So that's an important point. So if you're just keeping inventory information, that there's a sample in a box and it just has an ID and a date, there's no PHI on it, that you can store on your computer. You can put it in box.com. Maybe has, everyone has access to Box. That's fine. Encore actually resides in the health system with HSIS behind the firewall, so that's a fully HIPAA compliant system, but you need to have approvals to get into it. If you are saving PHI, you shouldn't be using Box. HSIS has a different system called ShareFile. It's not as easy to use as Box, but it works. And so we keep all of our PHI information on ShareFile, not on Box. So you just need to be careful about that. As long as you're password protected, you can keep it on your own desktop. You need to work with your IT group to understand what your needs would be if you need to keep larger data sets that contain PHI located somewhere. That's something that the Informatics Institute is acutely aware of. We're actually working with them. There was a program called UBright. I don't know if anyone heard about that last year, which is a pilot program for studies that are doing large clinical-based research with large data needs that are both clinical and research-based, trying to work through where are things stored, how are they stored, how are they accessed. And HSIS is finally working with UAB IT and UAB research computing to come up with solutions to make it easier, but still maintain compliance with regulations for data storage. We're still a little fragmented here, but it is getting better. But the informatics team is good at understanding what what the needs are, and they're happy to consult to help with that.
Any other questions? Okay,
Okay, thanks.
thanks very much.